Hello everyone, my name is Charles Wise, and I'm briefly going to go over my research conducted at Western Carolina University under Dr. Amanda Storm. So we investigated an uncharacterized protein in Staphylococcus aureus, as seen in the middle there. That is the protein structure. And so let's go over some background information about this, or the reason why. So some background about this research project is, of course, we are researching an uncharacterized protein in Staphylococcus aureus. This project is actually building off a larger project being conducted by the Center for Structural Genomics of Infectious Diseases, and what they do is they actually look for peculiar proteins that interest them. They image those proteins and they upload them to the database, and that's what we're doing. We're taking an entry from their database and trying to characterize it through bioinformatic techniques. And we chose 3UN6 because of the type of complications Staphylococcus aureus is known to cause, and 3UN6 had some nice pre-existing annotations that are really useful for building a stronger hypothesis using bioinformatic techniques. So let's get on to the methods of the research. So let's go briefly over the methods that we used for this research. So to identify a possible protein function, we used 3 assessments using bioinformatic programs and databases. So first, we're using 3UN6's sequence to compile predicted domain, motif, and residue features and figure out why these programs are coming up this way, what makes them similar to 3UN6. Second, we're using these other predictions from these programs and what proteins are most similar being predicted through UN6 and running a multi-sequence alignment and characterizing what features are similar or same. And then third, we're doing almost the same thing as we're doing in the second, but we're taking the tertiary structure and we're identifying those possible characteristics and those similar characteristics to these other proteins that are being predicted similar to 3UN6. So from our first assessment, we got back a lot of data that was useful. So using 3UN6's sequence, we got back that it was likely an ABC transporter. It was binding in the periplasmic space to the cell. It was the substrate binding protein unit of an ABC transporter, and it was transporting a trigonal planar molecule such as nitrate, bicarbonate, or sulfate. And so what an ABC transporter looks like is this image in the top right, and the SBP or the NRTA is what's being predicted. So it would bind nitrate in the periplasmic space and line it up to a transmembrane transporter that would then transport it into the cell. So here is our results from the second assessment. So we aligned multiple sequences with the same class of predicted ABC transporter as 3UN6. So NRTAs are nitrate binding ABC transporters, and CMPAs are bicarbonate. And we actually got some mixed similarities with both. But what we mainly got is that a lot of the residues that are important for nitrate and bicarbonate binding in these NRTAs and CMPAs are highly conserved in 3UN6. Not one is more similar than the other, which is interesting because you'd think one might be more similar than the other, but they kind of shared across the board. So on to the third one. So in our third assessment, we analyzed the tertiary structure of 3UN6 and its probable binding site. So what we found was 3UN6 had a highly conserved domain region. It had a structure that was very similar to an annotated NRTA protein. Its binding site was very similar to the NRTA protein. And using AutoDocVina, which is a bioinformatic tool for computational docking of a ligand, we actually found that nitrate was binding in the highest, or had the highest affinity score out of all the molecules we tested. So on to the conclusion of these results. So in conclusion, based on the 3 assessments, we think 3UN6 is the substrate binding portion of an ABC transporter. It attaches to the cell within the periplasmic space, and it's transporting likely nitrate into the cell. If you have any questions or want to learn more, because this is a very brief introduction, please look at our poster or come into our poster session. Thank you very much.